Okay, can somebody confirm you can hear me?
We can hear you.
Great. All right, before we begin, are there any questions? Today is March 4th, and we are supposed to be starting chapter 13. I don't think we're there. I think we're on chapter 14. Let me remind you, you have your last quiz this, tomorrow, Wednesday. This quiz will only cover chapter 8 and lab 9. There is no lab today. However, you should be working on your unknown notes, your unknown project. And let me remind you, your unknown notes pages will close March 7th. I think that's a Friday. And then the unknown project is due on March 8th, and that's Saturday. If you do request something on March 7th, please make sure that this will take only 1 day to incubate, because if it's a test longer than 1 day, most of the tests take 2 days, you will not get the results in time to turn in your project. Any question about any of that? Any questions about anything? Let me stop sharing the screen. I've got some grading done. There we go. Stop sharing the screen. I want to call up the grade book because then I'll remember exactly what I got graded. I've got a number of labs graded for the second time. Oh, I also got the extra credit study group graded up to about extra credit study 7. Let me see if I can scroll this way. Oh, come on. Yeah, up to extra credit study 7 has been graded. The lecture worksheets have been graded up until, well, I don't have lecture 8B graded yet, but I have lecture 8A graded. So I still have to grade lecture 8B and lecture 14. And I have labs 2, 3, 4 graded for 2 times. I'm working on worksheet lab 5 the second time. And then worksheet lab 9 is graded the first time. I probably did that earlier. Yeah, it's too late to turn that in for a second grading. So that was actually last week. I should have mentioned it earlier. Any questions about the grading?
I have one on extra credit study 4. It says this assignment does not count toward the final grade. Were you aware of that?
No, I wasn't aware of that. Let me see. It should give you the extra credit points, but you're saying it's not going to count?
Just on
Extra credit
EC
study
study
4?
4.
Let me look into that. Yeah, that's not supposed to be. It's going to count. And I just changed it right now.
Awesome, thank you.
All right, thanks for pointing that out. All right, any questions about anything? Let me get out of the grade book here, and then I'll share my screen again. All right, you should be seeing my screen. So last time I talked about the predisposing factors that can make someone more susceptible to getting a disease. Any questions about that? If not, let's talk a little bit about the development of disease. Once the host defenses are reached, disease development surprisingly follows a certain sequence, no matter which disease we're talking about. The sequence is similar for both acute and chronic diseases. First, there is an incubation period, and that is the period from when the person first becomes infected till the time the person first has signs or symptoms. So during the incubation period, you have no signs or symptoms. During the incubation period, you also have a small number of microbes, which will be increasing in number from when you first became infected. And then once the microbes reach a threshold number, the patient then will get their first signs and symptoms of disease. So that's the incubation period. Any questions about it? The prodromal period is the next sequence, which follows the incubation period, where the signs and symptoms are mild. Like when you first feel you have a sore throat and you ask, geez, do I have a sore throat coming on? And you have to think about it. And then you say, yeah, I've got a mild sore throat. So that's the prodromal period. The prodromal period ends when the signs and symptoms are no longer mild. And that is known as the period of illness, when the patient has the most severe signs and symptoms. And usually, this is also when the patient has the highest number of microbes inside them. So, both on the y axis, we have the number of microbes, as well as you can say the signs and severity of disease. The higher, the greater the signs and severity of the disease. The period of illness is followed by the period of decline. The period of decline occurs when the patient's signs and symptoms start getting better. Like when a person has a fever and then the fever breaks, that is the period of decline. Any questions about any of that? The period of decline continues with the number of microbes getting less, the signs and symptoms getting milder, until there's a point where the patient has no more signs and symptoms. So that's the period of decline, when the patient starts feeling better, and then when the patient doesn't have any more signs or symptoms. The period of decline is followed by the period of convalescence, and this is when the number of microbes in the individual are below a certain threshold, so that there aren't enough microbes in the individual to give the individual any signs or symptoms. But you notice that the individual still has microbes in them, and that's why it's called the period of convalescence. And that period will continue until the individual has no more microbes in them. So that's the period of convalescence. Any questions about any of these periods? Well, here's a question for you guys. During which period is a patient no longer contagious or communicable? The incubation period, prodromal period, period of illness, period of decline, period of convalescence.
I'm going to say period of convalescence.
Period of convalescence, that's a good guess, but you'll notice that the patient still has microbes in them, so they still can spread microbes to another patient.
So the incubation period is what you're saying,
Actually, in
or
the
no?
incubation period, the patient has microbes, so they are capable of spreading microbes. Admittedly, early in the incubation period, they're not likely to spread microbes, but late in the incubation period, they are likely to spread microbes. For many common diseases like colds and flus for adults, the patient is most contagious during late in the incubation period and the prodromal period. So for an adult, for many common illnesses, they are most contagious late in the incubation period and in the prodromal period. Even though they have fewer microbes, in an adult in the period of illness, when they have more microbes, the immune system is ramping up and it's attacking the microbes, preventing the microbes from spreading to other people. And I think that has something to do with our caveman ancestry, and that is for the adults, they did not want to spread the disease to their children. And so their immune system ramped up and made the disease less contagious in the period of illness than it was in the late in the incubation period and in the prodromal period. And the reason why it's more contagious here is the immune system hasn't ramped up by this time. It's too early in the infection. Now, for children, they are not this way. So any child underneath the age of 13, they are most contagious in the period of illness. And in fact, when I was a child, I think I was 12 or 13, I was not allowed into the hospital to see my brother, my younger brother, who got in a bicycle accident with a motorcycle. He was in the hospital and they said I could not see him because they didn't want children in the hospital because they were just too contagious. I didn't have any illness at the time, but they were just being precautious. They didn't want to spread disease in the hospital. Nowadays, the hospitals don't follow that rule. You can tell that's a long time ago. And they realize the social value, especially grandparents seeing their grandchildren, that it really does the grandparents good. And so they allow children into the hospital, even though they are very contagious in the period of illness. Any question about any of that? Now, that's a trick question. I won't ask a question like that on a quiz or the final, but it's just something to think about. And that is my mother, whenever she was getting better, she would say, "Oh, come on over and visit me. I'm no longer contagious. My signs and symptoms are better." And I would always tell her, "Mom, you're speaking to a microbiologist. I know better." Any questions about any of that? All right. So let's talk a little bit about the spread of infection. Oh, wait. I need to come back here and talk about the development of disease. Some diseases do not have all the periods. For example, sometimes I would be walking around feeling fine, not realizing I was going to become sick. I'm in the incubation period. And then the next moment, it feels like somebody hit me over the head with a sledgehammer. And then I came down with the flu. So sometimes with the flu, there is no prodromal period. You're in the incubation period, and then you're in the period of illness. And then there's some diseases where there is no period of decline or period of convalescence. In the olden days, like the 1980s, 1990s, maybe even the year 2000, I don't remember, when a patient got AIDS, they had the period of illness and then they died. So there was no period of decline, there was no period of convalescence. With my mother, she got shingles, and she had shingles from when she first got it till the day she died. So she was always in, I suppose, the period of decline. She was in the period of illness, but it got better. But she never got rid of the disease. She had it for the rest of her life. And that was at least 3 years, maybe 4 or 5 years. I don't remember. So sometimes you have a disease where it never goes away. Any question about any of that? Now, we're here talking about infectious diseases. Like if you get tennis elbow, that's another disease that never goes away. But that's from wear and tear on the elbow from playing tennis for a long time. All right, let's talk about the spread of infection. For an infection to spread, there has to be a reservoir of infection. So a reservoir is a source, a continuous source of disease organisms. Reservoirs can be living or non-living. Humans are reservoirs of disease, especially of human diseases. Example, AIDS and gonorrhea can only be obtained from another human. It is true, I guess, that AIDS can be in a chimp, but you're unlikely to get it from another chimp. And then it doesn't give AIDS to a chimpanzee. It just, I think it gives them the sniffles and then they get better. Um, so it doesn't give AIDS to a chimpanzee. And I don't remember, it only can survive in one of the chimps. There are two chimpanzees, two species. Humans can be carriers of disease, including humans which have an inapparent or asymptomatic disease or latent disease, like somebody who gets cold sore. When the cold sore goes away, they're in latency, but they can still spread disease when the cold sore comes out of latency and then forms another cold sore. And then you can be having an asymptomatic disease and then spread it to somebody else. I mentioned before the COVID-19, about 50% of teenagers, I think older children, and young adults will be asymptomatic for COVID-19. And the danger is they don't realize they have a disease, and then they can spread it to somebody older, such as their grandparents, where it can cause serious disease. There are also animal reservoirs. Some diseases like rabies and Lyme disease only comes from an animal reservoir. A zoonosis is an animal disease that may be transmitted to humans. Like rabies is really an animal disease, but when a dog or some other animal scratches or bites a human, the human can get rabies. But rabies is a dead end in a human, meaning the virus does not spread from another human. So once a human gets rabies, it's not going to spread to any human or any animal. Any question about any of that? And then there can be non-living reservoirs, like botulism tetanus, or two diseases that come from non-living reservoirs. An example would be soil, like tetanus, botulism, canned food, but there's other non-living reservoirs like water and lettuce. Now, contaminated water doesn't happen very much in the first world countries, but it happens all the time in third world countries. And then I'm sure you recall hearing about the recall of lettuce or spinach or something like that, contaminated with an organism which can cause a disease. Any question about the reservoirs? All right, let's talk a little bit about transmission of disease. There are three principal routes by which a causative agent of disease can be transmitted from the reservoir to a susceptible host. There's contact transmission, there's vehicle transmission, and that's where an object gives the disease to a healthy patient. Usually, the object gets the disease by touching either an infected patient or an infected animal, something like that, and the object gives it to the healthy patient. And then there's vector transmission. This is where an animal spreads the disease to a patient, like when an a rabid animal bites a human, that would be a vector transmission of the rabies. Vector transmission can also happen coming from a person, like when a mosquito bites a malaria infected person. The mosquito then gets infected by malaria, and then this mosquito can then bite a healthy individual, spreading the malaria by a vector. Any question about the three principal routes of the transmission of disease? All right, contact transmission has three types. There's the direct transmission, and that's person-to-person transmission requiring close association between an infected individual and a susceptible host. Like when you give a handshake, if somebody's infected, they can transmit the disease with the handshake. Kissing, another direct person-to-person transmission. Like when your significant other has a cold, you might hold off on doing the kissing because that can spread uh, disease. Another thing is normally you might kiss your children if you have children. If you have a cold, you might not want to do that because that would be direct person-to-person transmission. There's also indirect transmission, and this is when the person spreads the disease by touching a non-living object, which then touches the other person, spreading the disease. The non-living object, we have a special term for that, and that's called the fomite. Like when somebody is sick, and then they touch money, and then they turn it into the cash register, and then the cash register gives you the money, you can get sick from touching the money. Obviously, the cash register person can get sick too. When children are playing baseball, if somebody's sick on the team, they can touch the ball and then spread the disease that way. There's also droplet transmission, that's a contact transmission, and this is when transmission is spread being airborne droplets that travel less than 1 meter. So when a person coughs or sneezes, if the droplet transmits the disease-carrying organism and it's transmitted less than 1 meter, that's considered droplet transmission, and that's considered contact transmission. I'm not going to test you much on droplet transmission and airborne transmission because droplet transmission would be the cough or sneeze from about here to there, but beyond that point, it's not droplet transmission, even though it's from the same cough or sneeze. So once again, contact transmission can be direct, high-fiving, indirect, touching the baseball, or droplet transmission when it's spread in the air by less than 1 meter or up to 1 meter. Then there's vehicle transmission, and let's talk about the third one there. Airborne transmission is a droplet that travels more than 1 meter. So that would be from here on. It's the same cough or sneeze. You don't need to know about this because I think it's somewhat arbitrary, this 1 meter boundary. It is true you're more likely to get it within 1 meter than beyond 1 meter, but still, it's the same cough or sneeze. And so I'm not going to test you on that just because it's a little arbitrary what is a vehicle transmission and what is a direct transmission. And it's the same cough or sneeze. A vehicle transmission is when the disease carrying organism are transmitted by an inanimate reservoir, which can be food, water, body fluids, or an object. There's waterborne transmission, and that would be when water is contaminated by sewage. It doesn't happen very much in the first world countries, but in the third world country, this happens way too often. Okay. There's also foodborne transmission when somebody's sick and they touch your food, and then you eat the food, you can get foodborne transmission. That's why the people in the food industry, when they're sick, they're not supposed to work because of foodborne transmission. The trouble is that they usually take medication and feel better, have no signs or symptoms, but they can spread disease. So that's something to worry about. It's especially true if you're handling uncooked food. If it's cooked and you handle it before it's cooked, it'll be killed by the cooking, almost always. But if they handle it after it's cooked, particularly if the food is cooling down, it can be spread. And then lastly, with vehicle transmission, there's airborne transmission, and we already talked about that. That's wherein the droplets travel more than 1 meter from the reservoir to the new host. Any questions about vehicle transmission? There's also vector transmission, and that's where an animal carries the disease from the reservoir to a susceptible host. The most important group of vectors are the arthropods, especially biting fleas, ticks, and mosquitoes. There's two ways that vector transmission can happen. There's mechanical transmission. It's passive, like when the fly passively transports the disease-carrying organism on the feet. The fly can pick it up here, spread it to the hamburger here, and then the patient eats the hamburger, getting the disease-carrying organism. Any questions about mechanical transmission? There's also biological transmission, and that's where the pathogen reproduces inside the vector. So when a mosquito bites you, the pathogen can actually reproduce inside the mosquito, malaria. And then when the mosquito bites another person, they can spread the malaria. So mosquitoes and malaria is a very good example of vector transmission. Another one would be, what the heck is the name of that disease? Lyme disease would be another example of biological transmission. Any questions about vector transmission? All right, so remember there's three ways that the disease can be transmitted: contact transmission, vehicle transmission, and vector transmission. Let's talk a little bit about nosocomial infections. This is one of the most important topics you can learn if you go and work in the healthcare industry. A nosocomial infection is also called a healthcare acquired infection. Nosocomial infections happen as a result of a hospital stay or a hospital-like stay, like you go into the clinic, you go into a nursing home, and you pick up the disease while you're staying in the home or hospital or whatever. This is a disease that the patient did not have before they checked in to the hospital-like environment. It's estimated that 5 to 15% of all hospital patients in the United States acquire a nosocomial infection. That's 2 million people per year getting a nosocomial infection each year in the United States, and that results in 20,000 deaths each year in the United States alone. It used to be the 8th leading cause of death in the United States. I need to look that up and see if that's still the case, because COVID-19 may have bumped it down one place. So it's around the 8th leading cause. It's either the 8th or the 9th leading cause of death in the United States. About 1 in 10 hospital patients get a nosocomial infection. That's from the Centers for Disease Control. And about 50% of nosocomial infections are preventable. excuse me here. Nosocomial infections have a major economic impact on our society. You don't need to know the numbers. $9.8 billion in the United States. I think that's per year too. That was in studies done in 2013, but my point is it's just a major impact on American health and American, what do you call it, gross domestic product. Nosocomial infections result from the interaction of three factors. First, there are microorganisms in the hospital or hospital-like environment. Hospitals are major reservoirs of sick patients, and these sick patients often have communicable diseases, which can spread from patient to patient. Some nosocomial infections are normal microbiota that become opportunistic pathogens, infecting the patient. And then some nosocomial pathogens are antibiotic resistant, and then those are the worst nosocomial infective agent. So it's antibiotic resistant, so then the doctor gives the first antibiotic, it doesn't work, and then he gives the second choice, and the patient's getting sicker and sicker from the nosocomial infection. And then they realize, this patient must have a antibiotic resistant bacteria. And so they do the study to find out what antibiotics might work. And of course, the patient has finally died because the nosocomial infective agent, broke the camel's back. It was just too much for the hospital patient. Anyways, microorganisms are present in the hospital environment. You can't avoid them. They're in the air, they're on the objects, they're in flower vases. And then most patients are in the hospital for a reason. Oftentimes, these patients are compromised hosts, one whose resistance to infection is impaired by either disease, therapy, or burns. Many hospital patients have broken skin, broken mucous membranes. This will compromise the host, making them more susceptible to getting an infective agent. Burns, injections, ventilators, urinary catheters, the administration of anesthesia all make the host more compromised and then more likely to become infected. Like a burn, obviously, removes the intact skin, which would be a barrier to many infective agents. Any question about any of that? And then the patient can also have a suppressed immune system from either drugs, radiation therapy, burns, or possibly the infection itself, making the patient having a weaker immune system. And then they're more likely to get a nosocomial infection, as well as the fact that it could be worse because there's no or a weakened immune system to fight it off, but it just makes it more likely. So instead of like salmonellosis, you need about 500 organisms to cause disease. Well, if the patient is compromised, they need much less than 500 organisms to get disease. Maybe they only need 100 or 50, something like that. And then there's a chain of transmission happening in the hospital or hospital-like environment. The principal routes of transmission can be direct contact transmission, which can be patient to patient, like a patient in the hospital shakes hands with the person who comes into their hospital room. You'd have to take special care in special care units so that disease doesn't transmit from one patient to another. And then there can be a hospital staff to patients, direct contact transmission. So when you handle one patient, before you handle another patient, you should either take off the gloves, put on fresh gloves, or you should wash your hands in between patients. There's also indirect contact transmission, and that can happen via fomite, such as when somebody who's sick turns on the light and then you're in the same room, you want to turn off the light to go to bed, you touch the light switch. There's also urinary and IV catheters, contaminated surgical dressings. All of these can be indirect contact transmission. And then there's vehicle or airborne transmission, such as disease spread by the ventilation system. This has been a real concern in the hospital because it's well known that colds and flus can spread by the ventilation system. When we first had Ebola patients in the United States, because they went to Africa to help with treating Ebola patients, and then they did something wrong and they infected themselves with Ebola. These patients who came from the United States, most of them returned to the United States to receive treatment. And there was real concern that could Ebola spread by the ventilation system. At the time, they didn't know how Ebola was spreading. They knew it was spreading by bodily fluids, but they weren't certain, can Ebola spread airborne? And to tell you the truth, I'm not sure that they know that it cannot, but it rarely does, if it does at all, spread airborne. But there's real concern that could the ventilation system in the hospital spread Ebola? What some hospitals have done is if there's a patient which becomes quarantined, they're set up in a quarantine environment, and then they filter the air that is pulled in from that patient's room. Before it's put into the rest of the hospital. And usually that's set up like they have a special place where they put the quarantine patients. So the air coming out of that room is filtered before it's spread into the rest of the hospital. Any questions about any of that? All right. There are ways you can control nosocomial infections. And obviously, if you work in the healthcare facility, you should work to prevent nosocomial infections from spreading under your care. Certain procedures are generally implemented. You've learned aseptic technique now in microbiology. When you go into the hospital-like setting, you should always apply aseptic technique. You should always properly handle contaminated materials so that the infective agent doesn't spread. You should also have frequent and thorough hand washing. This alone has been shown to reduce the incidence of nosocomial infection by 50%. You educate the staff, the patients, and the visitors. In the hospitals in the 1950s and 60s, you would notice that the doctors proudly wore their stethoscope on a patient, and then they would listen to the heart of a patient touching the stethoscope directly to their skin. And then they would thump it onto their chest, and then they go on to the next patient, taking the stethoscope and putting it directly on the skin of the patient to listen to the patient's heart. There was no cleaning of the stethoscope. And in the 1950s and 60s, the number one nosocomial infective agent was Staphylococcus aureus, which was spread by the hospital, mainly the staff, simply by touching one patient's skin who had Staphylococcus aureus and then spreading it like with the stethoscope. The doctors quickly became educated, and so that stopped sometime in the 1970s. They then started educating their hospital staff not to spread nosocomial infection. And then they started the patients educating them. Wash your hands while you're in the room. If you have a cold or flu, after you blow your nose or sneeze into your hands, wash your hands before spreading it. And then lastly, they educated the visitors. Now every hospital, at least in the United States, has a large sign saying prevent the spread of disease, sanitize your hand, and there's hand sanitizer all over the place in the hospital, such as in the elevator, in the entrance to each room. And that greatly has reduced the spread of nosocomial infection by simply educating the staff, the patients, and the visitors. In fact, the incidence of nosocomial infection for the last 5 or 10 years is actually decreased by one half. So this is a little bit out of date here. Where is it? Right here? No, right there. 1 in 10 hospital patients, or 5 to 15%. It's no longer 10 or 15%. It's really only about 5% of hospital patients in America comes down with a nosocomial infection. So instead of 1 out of 10, it's now 1 out of 20. And that's largely because of education. There are some other ways to control nosocomial infective agents. You can look at the chapter notes, and I discuss those there. But you should really work to control nosocomial infective agents, because when a patient checks into the hospital, in the first place, they're not expecting to become sick. And then you don't want them to get a nosocomial infective agent while they're in the hospital. Any question about nosocomial infective agents? All right, let's move on. Epidemiology. Epidemiology is a study of where and when diseases occur and how they're transmitted in populations. The CDC collects and analyzes U.S. epidemiological information. Usually what happens is the local counties send that information to their county health department. The county health department then forwards that information to the state health department. I used to live in Oregon, so I lived in Washington County. So the doctors would send all that information to Hillsborough because that was the location of the health department for Washington County. And then they would forward it to the Salem, where the health authority is. And the reason why I'm talking about Oregon is I just know, like in Vancouver, I am sure that in Clark County, the health department is someplace in Vancouver, but I don't know where. I'm sure it's in Vancouver, though. I'm not 100% certain, but I'm pretty sure. But I'm not sure where the county sends it to. I don't know where the state of Washington health department is. Usually it's in the capital, but Olympia is much smaller than most other cities and in at least most other large cities in Washington. And I just don't know where the health um, office is in the state of Washington. If somebody knows, you can pipe in. I've never had a student tell me, but I just don't know. I should look it up sometime. And then they send that information. The health department for the state sends it into the CDC. The CDC then collects it and analyzes the U.S. epidemiological information, and they publish it in the Morbidity and Mortality Weekly Report, frequently called the MMWR, weekly. There was one exception, and that's when Trump took office and then put an immediate freeze on the federal government. The Morbidity and Mortality Weekly Report was not printed for that week. That's because they had a budget issue, and I don't know. They were supposed to freeze things, and that's one of the things they froze. And then you can find that at www.cdc.gov, which I used to be able to find it very easily. It's not so easy to find anymore. Well, I can do a search here. MMWR. There we go. There it is. Morbidity, Mortality Weekly Report for the Center for Disease Control.
I also Googled Olympia, Washington is where the Washington State Department of Health's headquarter is, and they said that they also have one in Tumwater as well, which is the Judicative Service Office in Tumwater.
What's a judicate?
I'm not sure, but that's what they say.
Okay. Well, thank you. I'll have to make a note of that. So it is in Olympia.
I would have thought it was in Seattle, even though it's not the.
It's not the capital, yeah.
Yeah.
But it's the biggest city, so that's why I didn't know in Washington. And like I said, I knew in Oregon, but I just never knew. I never ran into it in Washington. All right, thank you. Anyways, that's something like that. Notifiable diseases are diseases that physicians are required by law to report to their county, which then reports it to the state, which reports it to the Centers for Disease Control. So notifiable diseases, they can be different in each state. Most diseases, every state will report them, like common diseases like flu or obviously COVID-19. These are diseases that must be reported. Syphilis, gonorrhea, they must be reported. The incidence of malaria that has to be reported, even though malaria isn't common in the United States. It happens mostly to people who travel outside the United States to the tropics, pick it up in the tropics, and then bring it back to the United States. However, in the Gulf states, as well as the southern states along the Atlantic, we do have the mosquito, which can spread malaria. So every once in a while, malaria is brought to the United States from somebody traveling outside of the United States, and then the mosquito bites that patient and then spreads malaria. That happened a few years ago in Texas and Florida and one other state. I think it was one of the Carolinas, I don't remember which, where malaria was actually spreading in the United States. Fortunately, the winter happened, killing off not all the mosquitoes, but some mosquitoes, and it killed off the malaria, and then malaria stopped spreading in the United States.
I know here in Washington, whooping cough is a notifiable one. My son had whooping cough and pneumonia, and it was going around all the schools, and I had to do an interview.
Yeah, there's many of the childhood illnesses which are notifiable. Measles and mumps are two examples, as well as whooping cough. All right, let's move on to epidemiology. Morbidity is the incidence of a specific notifiable disease. The morbidity rate, the number of people affected in relation to the top total population in a given time period. Mortality is the deaths from a notifiable disease. The mortality rate, the number of deaths from a disease in relation to the population in a given time. Any question about any of these terms? Any questions about chapter 14? If not, let's move on to chapter 13. Viruses, viroids, and prions. This slide is showing you both the major goals and a rough outline of viruses, viroids, and prions. Obviously know the terms. 2, understand the characteristics of viruses, their structure, and their classification. 3, understand about viral multiplication. And 4, understand about prions and viroids. Any question about the major goals and the rough outline? The only goal a virus has is to replicate. How they do that is they invade a host cell, hijack the host cell, manipulate it and force it to replicate the virus. And then the virus escapes from the cell. Let's watch a little movie on bacteriophage replication. That's a bacteriovirus. Little video, I should say.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any question about that? Okay, so know some of the general characteristics of viruses. Viruses are inert when they're not within a host cell. They display little or no metabolic activity outside their host cell. Viruses do require a living host cell to multiply. So you can say a virus is an obligate intracellular parasite. They multiply within a living cell by using the host cell's enzymes, nucleic acids, amino acids, and ATP. Viruses have no ATP generating mechanism. So for their ATP needs, meaning their energy needs, a virus will use the ATP that is inside the cell. A virus does contain a genome, but this genome can be either DNA or RNA. It is never both DNA and RNA. It's one or the other. And a genome that's DNA is normal. That's what our cells have. In fact, all cells have DNA for their genome. A genome that's made of RNA is unusual. But our viruses can have a genome, depending on the species of virus, can have a genome which is RNA. A virus always has an inner molecule, DNA or RNA. Around that is a protein coat surrounding the nucleic acid. And this is a virus showing the protein coat here. And this part of the code is cut out so you can look inside the virus. Some viruses also are enclosed by an outer lipid envelope, which is outside the protein coat. Not all viruses have this outer lipid envelope. The host range of a virus is the range of host cells that the virus can infect. This is determined by host-specific attachment sites that are called receptors. And these can be molecules on cell walls, on flagella. They're always a molecule on the outermost portion of the cell. And then cellular factors necessary for viral multiplication, as well as the molecule of the virus, which binds to the receptor. I blow this up. So here we have a receptor, a virus can bind to it, and there we have the viral protein coat, and it has called adhesions or ligands, which are molecules on the virus that can bind to the receptor. Most viruses only infect cells of one or a few species, usually only a few cells of that species. A classic example is AIDS, can only infect, well, I shouldn't say only infect, mainly infects human T cells, macrophages, brain cells, and a few other cells. HIV, that virus cannot infect skin cells, muscle cells, or bone cells. So the majority of the cells in a patient cannot be infected by HIV. And then HIV only infects really two species, humans, and one of the species of chimpanzees. There's two, only one of them can get HIV. And when they do, it does not cause AIDS. I think the chimpanzees gets the sniffles and that's it. Where was I? There are some exceptions where a virus can infect many different species. Like a bird flu, it's well known to hit many birds, and there's many different species of birds. And to my knowledge, it can infect any bird, but I'm not positive of that. So we'll just say any bird or close to it. And then it can infect many mammals. I don't know if it can infect cold-blooded animals, but it definitely can infect many different warm-blooded animals, bird flu, including humans. And with most mammals, as well as humans, bird flu may be lethal. But most viruses only infect one or a few species, and usually only a few cells of that species. And the reason being is the specific binding of the ligand or adhesion molecule on the virus binding to the specific molecules of a receptor, which is a molecule on the host cell. Comparing the size of a virus to other cells, this one here is a little bit of a red blood cell. And there you see the very largest virus, longest virus. And there is E. coli, which is a little bit longer than most bacteria, a little skinnier than most bacteria, but most bacteria are around 1 to 2 micrometers in diameter. And Ebola is the longest virus, but not the largest virus. The largest virus we're going to talk about is megavirus, and it has a larger diameter than Ebola, because Ebola is pretty skinny. Longer, but it's skinnier. And then so that makes the largest volume, megavirus. Most viruses are smaller than this. Megavirus is actually approaching this size a little bit larger than the size of the smallest bacteria at 0.4 micrometers in diameter. Most viruses are smaller. The smallest virus you know of is the polio virus, which is, I believe, 30 nanometers. The smallest virus gets down to 20 nanometers or 24 nanometers, like bacteriophage MS2. So there's a few smaller than the polio virus, but not much smaller, just a little smaller. So the polio virus is the smallest virus you've probably heard of. I guess what I should say here is your average virus is about 1/10 the size of your average bacteria, which is about 1/10 the size of your average eukaryote cell. When we're talking about the viral structure, it's important to realize that a single mature virus is called a virion. This is a single mature complete infectious virus particle. Virion is a term used synonymously with virus, but virion is usually used when we're talking about the newly replicated viruses. They have a nucleic acid inside them as their genome. That genome can be either DNA or RNA, but never both. The DNA virus can be double-stranded, and that's pretty normal. Our cells are, for the most part, double-stranded DNA. But some viruses have a genome which is single-stranded DNA. Single-stranded DNA is not unheard of in our cells. Whenever our cells are replicating or transcribing, that region of the DNA is single-stranded. Viruses' genomes can be double-stranded RNA, and that is unheard of in a eukaryote cell. Eukaryotes do not have double-stranded RNA. But viral genomes could be double-stranded RNA, as well as they could be single-stranded RNA. Now, RNA as a genome is unheard of. RNA in our cells that's double-stranded is unheard of, unless you're infected with an RNA virus. The RNA genome can be single-stranded RNA, and if it's single-stranded RNA, it's important to state whether it is a positive single-strand RNA or the negative single-strand RNA. The positive single-strand RNA genome can be read by the ribosome as messenger RNA, meaning AUG will appear in the positive single-strand RNA molecule. And then AUG will not be found in the negative single-strand RNA. Instead, this will be the corollary to the positive strand. So instead of A, you have U. Instead of U, you have A. Instead of G for AUG, you have C. Any question about any of that? Viruses can have linear or circular genome. A linear genome is what our cells are, like on the X chromosome. That's a linear DNA molecule. Well, viruses can have a linear DNA or RNA genome. They can also have a circular genome, and that's common in bacteria, where their genome is circular, and viruses may have a circular genome. It depends on which virus we're talking about, which species, but viruses are either similar to a prokaryote cell or they're similar to a eukaryote cell. And it just depends on what the virus is. Some viruses, like influenza virus, have their genome in several segments. So it's not in one segment. The genome is split into many different segments. And that's common in our cells, where we have each DNA molecule on one segment that we call a chromosome. We don't call virus DNA or even RNA chromosomes because that term is used for a DNA molecule that has proteins in and on the DNA molecule. Any question about any of that? All right, once again, we're looking at a virus. That's a central molecule, and then that's surrounded by a protein coat. You can't have the envelope around the protein coat, depending on the virus. The capsid is the protein surrounding the genome of a virus. It is composed of capsomeres. Let me blow this slide up. There can be one capsomere in the virus. Let me blow it up even further. Like this virus has only one capsomere, and that's one there, that's one there, that's one there. So it's a whole bunch of capsomeres which make the protein coat of the virus. But in this case, for this virus, it's only one protein making up the entire capsid, virus DNA, the protein molecule around the DNA. Let's shrink that down. If there's no envelope around the virus, we call it a naked virus. So this right here would be a naked virus. If there's an envelope around the virus, we then call it an enveloped virus. And this is an enveloped virus right here. In fact, right there you can see the genome. Right here you can see the protein coat, the capsid. And this would be the envelope. Envelopes tend to be a lipid bilayer. There are proteins and carbohydrates in that lipid bilayer, but it's mostly one molecule, and that is a phospholipid. The envelope may be covered with glycoprotein spikes. These reddish purple things are the glycoprotein spikes. These reddish purple things serve as antigens, meaning that when the host realizes it's infected with the virus, it will make an response to the virus by causing antibodies to be made, which will then bind to the spot that was initially discovered by the I don't know, the probe or the scientist. These glycoprotein spikes are the furthest molecule out, and I already mentioned that the furthest molecule out usually are the points of binding for the virus to the receptor of the host cell. But these furthest spikes, or the furthest molecule on the outside, also contains the antigens, which are the molecules that the host immune response will respond to. The whole immune system will respond to these glycoprotein spikes because that's the furthest molecule out. And what molecule is the furthest molecule out on this one? It's going to serve as the antigens for this molecule, or this virus. All right, it's the molecules of the capsid. They're the furthest out, and this capsid will be the ligand or adhesion of the virus that will actually bind to the receptor. And then the capsid here will also be the furthest molecule out. So the patient's antibodies will attack the molecules in the cell membrane because that's the furthest out. Any question about any of that? All right. When we're talking about viruses, it's important to talk about their general morphology, that is the shape. Viruses are classified on the basis of their capsid structure into three major groupings. The helical virus, shown here, where the DNA and the capsid both take on the helix, the coral staircase. Viruses can also be grouped as a polyhedral virus, such as one here. This has been cut out to allow you to look inside, but other than that, this is a polyhedron. And there's a polyhedron here. This is an actual electron microscopic image of a virus. And there's the protein spikes. You can see those. Not many of them, but you can see those in the plane. All right, any questions about the viral structure, the capsid, the envelope, the glycoproteins, or the viral structure? I guess I haven't talked about the third one. Let me talk about the third one. And, third virus is a complex virus. Virus can take on any shape, depending on the species. This species takes the shape of a lunar lander. I think that's the Apollo or Soyuz mission. No, that's the Apollo mission. And then this is the moon lander. And the virus will only bind right here in the bottom of the feet. So those are the molecules of the virus that bind to the receptor of the host. And remember, the molecule of the virus, which is binding to the receptor, as well as the receptor itself, these are specific molecules, and the host cell may change them, like if the bacteria are infecting the egg, the mother may attempt to alleviate that by having the immune system mount a response against the virus. Anyways, this is a complex virus taking on many different shapes. This is the artist's rendition. That's an actual electron microscopic image. And we're showing you the T-even family of viruses, which include T2, T4, and T6. Bacteriophages, which are another term for bacterioviruses. Viral infection and the host immune response. Infection with a virus can stimulate the host to produce antibodies, which will then recognize and bind to the infective agent, in this case, the virus. In a virus, the antibodies can bind to the viral surface proteins, and these are antigens. And the virus can either inactivate the virus or can directly attack the virus, or stage the virus to be eaten by the host immune system, meaning flag the virus to be eat me by the host immune system. We'll talk a little bit more about that later. Some viral proteins can change or mutate frequently, so different viral strains can therefore infect the same individual more than once. A good example of that is influenza virus, where it's changing its surface molecules, surface antigens, as well as changing other aspects of the virus. And that's why when you get a vaccine to the flu, they say you must get a vaccine again about every 3 years because the immunity, so this would be time down on the x-axis. I don't know if I can write this or not. Kind of. The y-axis is percentage of immunity. I'm just going to put up here percent. Meaning how well you respond. I'm going to just type that immune. IMM. So when you're first vaccinated, your percent immunity will be very high. Not quite 100%, but close to it, or at least 90%. And over time, that immunity is going to drop. Once it drops below a threshold, I don't know what that number is, but once it drops below a threshold, they say you're no longer immune. Obviously, your immunity has dropped from, I don't know, 90% down to something like 30%. It doesn't quite die like that either. It's going to decrease slower with time. Something like that. And that's why they say every 3 years you have to get a flu vaccine, because if you got it more than 3 years ago, your immunity is too low. You're not likely to be protected against the virus causing the flu. But if it's above this threshold, then at least you have some immunity, which will protect you. If it's really high, you'll most likely be 100% protected, but realize 100% of the people will not be protected 100% of the time. In any case, that'll never happen. It never gets higher than about 90%, 95% of the people having an immune reaction. And sometimes that immune reaction is kind of weak, in which case you could get the flu. It'd just be milder than the normal flu because your immune system is already fighting it even before you got signs or symptoms. Any question about any of this? Another example is COVID-19 does change its molecules pretty quickly. And that's why with the flu, you have to get a vaccine at least every 3 years. They recommend every year because, as you saw in my graph, the longer you have, the less immunity you have. And they like to keep the immunity high, higher than 50%, let's say. And to do that, you need to get the flu shot about every year. And then COVID-19, similar. It changes its proteins fairly rapidly. A new strain will then come out and you will lose immunity to it, which, if it's very weak, it may not even prevent disease. Fortunately, most viruses don't follow this pattern. Like we still are given the polio vaccine out. One of the polio vaccines was developed in 1954, the other in 1958. That's obviously a very long time. What is that, 75 years? Something like that. Close to 75 years. And the virus has not changed. The vaccine is still effective. And that's a good thing. That way we don't have to do a new vaccine every year, which you have to do for polio, not polio, the flu and for COVID-19. Any questions about any of this? If not, I'm going to stop here. There is no lab, but if you have questions, I'll be logging in for 15 minutes from 6:30 to 6:45 to answer any questions. When the questions are all done or 6:45 comes around, I'm going to log off the lab. All right, any questions? All right. I'll
Thank you.
lecture. You're welcome.